Hi everyone, I'm Andhika from the University of Melbourne down in Australia, and today I'll be giving you a quick talk about my PhD project, which is trying to understand how invasive species respond to the challenges of novel environments. So just to start off as a very basic concept, for species, for us to consider species invasive, it has to be moved from whatever we call the native range to the invasive range. And this simple movement entails moving only a subset of the individuals that make up the whole species population. So in this conceptual diagram I have here, you have the native range and some level of diversity there represented by all the plants with the different colors. But when you move it to the invasive range, only some of those individuals will get transported. And this very simple fact results in two main things, which is first, you have fewer actual individuals, and second, you have reduced genetic variation. Why is that important? Well, that's important because the reduction in genetic variation comes along with it the introduction to the novel environment. And so during the invasion process, you have all these factors that are changing or varying. So the framework I've made to that kind of underpins my whole project is the idea of separating three components. So you have phenotype, genotype, and environment. And all these three components can vary, and they're all interconnected to each other. So for example, the relationship between phenotype and genotype would be genetics, or changes in phenotype in response to environmental variation would be phenotypic plasticity. And then also you can have differences in the way genotype and environment are distributed. So under this framework, the driving questions of my research at the highest level is really, how do these components influence each other? What happens when I change one component, or what happens during invasion when it changes? And why do we want, and then ultimately, can we predict the consequences of these changes? Now, prediction here is kind of key because practically, if we look at invasive species, we don't want them around. So to be able to predict would give us a sense of being able to predict if and when invasions could occur, and we could take on different management strategies to combat that. But also evolutionary, and in the sense of like furthering evolutionary biology, it's really quite interesting to kind of predict what changes might occur. So this study, I'm doing this on common ragweed, which is actually native here in North America, but down under it's invasive. So ragweed is an annual plant that's most notable characteristic is it produces really allergenic pollen and is a big contributor to hay fever. So for the purposes of this study, we're considering three different ranges. The native North American range, the European range, which is also invasive, but has been invaded much earlier, and throughout ragweed is a lot more widespread, and the Australian distribution, which is a lot more restricted. Now these characteristics make ragweed, I believe, a good system because firstly, the different distributions kind of represent different phases of invasion. So you have the native range and you have a fully invaded range in Europe where it's a lot more common and widespread. And then you have this restricted range in Australia where could it be a lag phase? Could it be its final distribution? That's kind of still one of the open questions that I'm actually trying to answer. So throughout the rest of this talk, I'm going to be using data that was already done way before I started this project. It's basically results from a common garden experiment where plants sourced from these three ranges were grown in the same garden. We have 860 plants that were both phenotyped and genotyped for different traits, and up to 158,000 SNPs, depending on, just because over time we've had better genomes and better alignments. So the first thing I want to discuss is looking at the relationship between genetic and environmental variation. And so here what I did was I took the genetic data and I identified 3 genomic clusters using an admixture program, creatively named cluster 1, 2, and 3. And the distribution of these clusters, what we found was actually non-random, that certain genotypes, certain clusters favored certain environments. So you can see in the top 3 panels for the 3 clusters, where each, from the populations that were sampled, that belong to each cluster, and then the larger circle is kind of the centroid, and what we see is kind of cluster 1 is in the west, cluster 2 is in the middle, and cluster 3 is in the east. If we look at where these plants were found, and we took the environmental conditions at these locations, and we created species distribution models from them, and we use those models to predict habitat suitability in Australia, we can see that they have different potential distributions. So in the bottom 3 panels, you can see, for example, cluster 1 has a potential distribution that's kind of further south in Australia, whereas cluster 2 and 3 are further up north. And that just highlights one of the things is it's not just about, during invasion, it's not just about bringing things over, but it matters kind of the identity of what you're bringing over and where they end up, whether you succeed or not. Another thing I was looking at is the relationship between phenotypic and genotypic variation. So the first thing we did was kind of create structural equation models to look at the trait correlation networks. And the main takeaway here is, we looked at 5 different traits in this case, so 2 life history traits and 3 morphological traits. And the main takeaway here is really that the traits differ in the way that they're connected to each other depending on the range that you're considering. Another interesting one is also that in Australia, the time that the plants flower, so Australian plants flower way later, but that actually has a negative relationship with the total fitness. So that's interesting because, we look at traits because that's the actual physical thing that's in the environment, that's exposed to pressures. So that's a measure of success. So if we look at how these traits are structured, the idea is we could measure maybe evolutionary potential, or in the case of invasive species, invasive potential about how successful they might be. But the thing is that because these because the way these traits are correlated differs so much between the ranges, what ends up happening is that the phenotype-genotype associations are actually inconsistent. So here I'm showing the results of genomic prediction models where I've tried to predict the trait as a result of using SNPs and then a genomic similarity matrix to account for population structure. And what I found was that models for all the traits were really only good at predicting within the same region. If you try to use a model that only saw plants from North America and you try to predict traits of European plants, the model prediction accuracy is just horrible. And you have to remember as well that this is all data from a common garden, so it's not an environmental effect, it's strictly differences in the way traits and genes have been associated. So that's it so far. You'll notice I haven't really talked about phenotype environment. So we've actually done growth chamber experiments there, and we do see differences in trait expression value in different environments. But that's kind of expected. We expect these plastic traits to respond to environmental changes. What I'm working on right now is we also have some field survey data, and the biggest challenge has actually been kind of to try and integrate all those 3 components together, so genetic, phenotypic, and environmental variation in kind of a temporal framework to model the population dynamics. So given a level of genetic variation and the conditions at that environment, what happens year to year? Do plants flower earlier? Do plants flower later? And that's really an open book. If anyone has any ideas in that space, I'm open to hearing your thoughts. So the main takeaways from my work is basically that this invasion process is really something that challenges our fundamental assumptions about how phenotypes and genotypes are correlated with each other. So something I found a lot is this tendency to kind of think of an invasive species as just a species, as just like individuals that are found elsewhere, but that's not really what we're seeing here. We're seeing that even in the short time scales that ragweed's been transported, the trade structures can really change, and it can be really quite complex to try and predict it. And I think that's probably why predicting invasions has still remained such a challenge in the field. So yeah, I'd like to thank my supervisors and my 2 labs in Australia. Yes.
How do you quantify and measure the total fitness?
Okay, so that wasn't something I did, but it was the total reproductive biomass of seeds and I think male flowers added together.
So the proxy, you use the proxy.
Yeah, as in, yeah.
The male reproductive.
And female, yeah.
Male and female
Yeah.
reproductive. Why do you use mortality and survival?
Because I think they're annual plants, so that they die at the end of the year anyway.
Oh.
Yeah. Yes.
I had a question about the geographic distribution of the clusters you found.
Yeah.
So it looks like the North American distributions are very overlapping. How did you end up with such different climate distributions in Australia?
So they do overlap. It's a bit hard to parse out here because of the point sizes, but although they overlap, each population might have a mix of cluster 1 and 2 and 3, but those numbers aren't equal. You can't see here. So for example, one of the populations might have 90% cluster 1 and 10% cluster 2. And so when you're fitting the model, it biases it towards preferring certain habitats because there's more observations with that environment. And then when you translate it to predictions, that changes the results.
Okay.
Yeah, so it's not fully one-to-one, yeah.
But thank you very much for coming here. I'm Chris Knight from the University of Manchester in the UK. What I'm going to be talking about is quite a big soil microbial. There we go. That's what I'm going to actually start. But it's great to be actually in quite such a diverse session. But I think there will be parallels between what we've just heard and some of what I'm going to talk about, which is a big microbiome study. Big both in the sense of lots of data on the left, and also big in terms of continental scale. In this case, the continent is Europe. And I'm actually going to start with my acknowledgements because this is very much was the brainchild of a grant that Francisca de Vries brought together with people from across Europe. These are the people who dealt with all the data, and there were individuals in all those different countries who were responsible for collecting samples. So while I'm the lead author on the paper, and I'm hoping the paper's going to be coming out pretty soon, data's out there already if anyone wants to play with it. It wouldn't have happened without any of all these people. And what we did, we were coming together to address the question of whether we can generalize and predict about the responses to extreme climatic events. So it's not a giveaway that the answer to this question overall I'm going to give is yes, we can generalize and predict, but actually our ability to generalize and our ability to predict are not necessarily the same thing, and there's some subtlety in what we found. Now, I'm an evolutionary biologist, but actually most of the time what I'm thinking about is going from the bottom, the biochemical details and models and experiments evolving in the lab, and trying to figure out how we can think about evolution. And I just put a plug in here for my PhD students who's got a paper which has just come out this week, who's going to be giving a talk from going from that direction, and actually thinking about constructing the really simplest communities of just two different genotypes interacting together. But I'm going in the opposite direction, and it does feel sometimes that we're sort of going in opposite directions, and that that bottom-up stuff, what I'm going to be talking about now is very much top-down, but it's been great, particularly, even potentially within this single session, that those two things might at some point start to meet in the middle a bit better. So we understand what's going on in these really complex systems, because we need to know what's going on in these complex systems. As environments warm, these kind of extreme events are becoming much more common. So the frequencies of droughts or high temperature events. And what we're going to do is think about the community responses to those events, because we want to understand, and we want to be able to predict what those responses are going to be like. And inevitably, those are going to be complex mixtures of intrinsic and extrinsic effects. The soil, if we're just going to be talking about soils, those systems are highly variable. The properties, all kinds of things that the ecologists working with on this project spend decades working on, how the relationships between pH and organic matter and how climate has shaped these communities. And that goes for the organisms as well, the kind of traits they might have. So we're going to do an experiment imposing these kind of perturbations and look at some rather basic sort of predictions. So we might expect that we're going to impose different climate extremes. Maybe there are common themes of what happens when you stress a community, but maybe there's a core of changes that we can say, well, this is what's happening in what happens when you drought, when there's a drought hits a community or heat hits a community. And so therefore we might want to expect to see parallel effects across different soils from different bits of a continent, where we can say this is that core response. But we also want to see whether that's in how that affects function and think about those functional genomic changes. We might go a little bit more subtly than that and think that, well, okay, if these communities have been evolving in a hot environment where heat events are more common anyway, maybe they are going to be more robust, so more resistant to heat, and bounce back, so more resilient, better when they are perturbed by a heat event, and maybe similarly for some of these other events. But those are the kind of hypotheses we're going in with. And what we're actually going to do is a common garden manipulation study. So we've just heard about one of those, but we're going to take soils from all these countries, which are representative of different biogeographic areas within Europe. We're actually going to take 3 different soils from grasslands in each of these places. And we took them all back to Manchester into little pots like that, and perturbed them for a couple of weeks using, drought, flood. I can give the details if anybody is interested, but importantly, there's a control samples for which we didn't do anything, and following the change of those communities anyway, and followed them for the course of a month. So I'm actually going to focus on the beginning and end of that recovery period. So we did a load of sequencing. Some of that's amplicon sequencing, looking at the bacteria and archaea in one lot, the fungi in another. And actually the metagenomics data I'm going to present, I'm actually only going to be thinking about that in terms of the functional genes. So what are the genes associated with particular traits that we see changing? And to cut to the first clear result, I'm not going to... This plot doesn't contain any information about the treatments that we did at all, nothing about the heat, but they're all in there, because what's blatantly obvious when we look at this is the community composition spread out in an MDS plot, is that the country is the wildly overriding feature of this data. So those pink blobs are all from from different grasslands in Greece, and whatever we do to them, they still look like a community from a grassland in Greece. They do not start to look like anything else very much. And so, for instance, this is my token gesture towards my actually collecting data here. I mostly did the analysis, but there I'm collecting a soil sample from a grassland, and just looking at that picture, I certainly would not know which of those countries that came from. But I would be able to tell the difference. I'm not a botanist, but I might be able to tell the difference between the vegetation in that field and the vegetation in the field behind. And actually, the grasslands we chose looked to me as a layperson very different, and yet they were much more similar when it came to the microbial communities. So certainly, as far as this data is concerned, everything is not everywhere. These are very distinct communities we're talking about. So these continental scale spatial effects are strong, and that's true for what the data I've just shown, but it's also true for the fungi, and actually for the genes that we have in there. So the colour is very highly differentiated among those community compositions. So, but can we actually then see anything about consistent changes? Well, actually we can. So if we go in with a sort of discriminant analysis approach to try and say, well, what changes are associated with these particular perturbations? And what you can see is that there is an effect of heat, quite distinct, going out to the right along that first axis, and that's quite distinct from the other three. But actually, the effects of freezing and drought are actually almost indistinguishable. They're going in very much the same direction, and that makes some biological sense, because what we're actually seeing is what that seems to be saying is that what really matters is water. That freezing and drought both restrict access of these communities to water, and therefore when we restrict that, they change in the same direction, which is largely in the opposite direction from when you add water. So the flooding goes in the other way. And we're seeing that in the bacteria and archaea, but we're also seeing it in the other data as well. But one thing you will notice, looking at any of these plots, is that the numbers on those axes, which are the percentage of variance explained, are tiny. I mean, even if you add them all up together, it's only a few percent. So have we done this massive experiment in order to find some tiny effect? Well, actually, the more massive the experiment is, the tinier the effect. So we can subsample this data, and if we've done it in one soil, one country, with 3 different soils from there, we'd have been able to explain maybe almost 10 times as much of the variance due to those perturbations. And we wouldn't have been wrong, we just wouldn't have been seeing the specific things going on that are generalizable across the response of all soils. So as we add the number of countries, we start to go down to some very small amount, so 3.5% in this case, when you actually add up all the possible axes. But that is where we're sort of plateauing out. We're asymptoting down to a low amount of variance with a high degree of certainty about what's actually in there. So it is possible to generalise, but only with a small proportion of variance. So what is it that's actually in there? So this is what happens when you start to peer into what's going on in those individual categories. These are a whole load of different functional categories. And what we mostly don't see too many things going across here. So each column is one of these perturbations. The one which stands out from there is that red line going across, which is dormancy and sporulation. So basically, organisms which are good at becoming dormant or sporulating generally do better when doing perturbation. So that's the kind of sort of generalized stress type things. Under heat in particular, we then see some specific reductions in organisms which have lots of clearly specialized to growth. So things like carbohydrate metabolism genes and protein metabolism, those kind of things are generally reduced. So heat in particular is shifting from growth to survival. Now I haven't got time to go into it, but actually it turns out that more generally, if we then look across the communities, we see different things. That there are these more copiotrophic communities that sort of boom and bust, that are good at, have many of those sort of growth genes, proteins. And we've got more oligotrophic communities which have many more of the sort of the subtle metabolic genes, the nitrogen metabolism, sulfur metabolism, things about virulence and killing each other. So, oops, there we go. Yeah, so we've got, and those are more resistant or resilient to specifically, they respond differently to heat and flood. That's the resilience and bouncing back. That suggests that actually that even though what we can generalize is quite small, maybe what we can predict is larger. Because these oligotrophic communities are from wet, acidic, cold places like the UK, and the copiotrophs are from hot, dry, more alkaline communities. And actually, if you put a number on it, about 60%, just under. And for that final hypothesis, actually, looking into that model, that temperature there is the climate temperature, so where it's from. So things from high temperature places tend to be more resistant, so tend actually to deal better with perturbations which are similar to the climate they've come from. And the same is true for wet soils and dry soils. So actually, that hypothesis that is mostly correct. So we can actually predict quite well what's going on. So leave that summary, but yes, there are different effects, but it's really small, but it is predictable, especially for heat, and particularly hot, dry climates are responding differently or better to heat. I'll stop there. Thank you. Any questions? Sorry, yes, you're back. Yeah, so thank you. So actually, that's sort of what we're doing in that model, where we can explain 60% of the variance. That's taking into account that climate where it's coming from. Now, actually, having done that, we don't actually get much more explanatory power from what's left. So it does work, but actually, so because that huge spatial effect. Actually, though, it is consistent. So I could have actually painted that first NMDS plot by the pH of the soils, and that would have made it clearer, because they are very much distinguished by those biogeographic features of the soils. Thank you. Anything else? No, thank you.
Okay, so hello everyone. Thanks for coming to my talk, and I'm pleased to see some faces that I talked with at my poster session yesterday. So I'm a PhD student in the lab of evolutionary and adaptive physiology in the University of Namur in Belgium, and I'm pleased to answer the following question with you today. Can DNA methylation predict the age of Cryptolebias marmoratus, the mangrove revolus? So, as a quick context, I would like to underline with you the importance of having age data. Indeed, age is definitely a key parameter to understand ecological and life history traits, whether it's at the individual or the populational levels. Traditionally, we were estimating age using the growth mark in hard structure, such as the otolith or the vertebra. Nonetheless, in addition to require trained staff and take loads of time, these techniques are actually lethal, and therefore they cannot be applied in every context. Just in the last 2 decades, we started to try several biological predictors to be used as an age estimator. So far, it turns out that DNA methylation seems to be the most promising one. DNA methylation is a stable epigenetic mark that plays an important role in gene regulation. So it consists of the addition of a methyl group on a cytosine guanine dinucleotide that I'm going to call after CPG. Along with the epigenetic drift, where DNA methylation gets altered as a function of the time, we also observe the epigenetic clock, which is change in DNA methylation that are correlated with the age at specific CPG sites. And that's what I'm interested in here. I'm working and developing that epigenetic clock on my model species, Cryptolabia marmoratus, the mongrel rivulus. So it's a killifish species from the family Rivelidae. Why am I working with that species? Well, it's a unique vertebrate that got a mixed mating system, where we have hermaphrodite individuals that can internally self-fertilize, giving isogenic lineages in the wild, but we also have males that can cross-fertilize with the hermaphrodite, giving us then a certain genetic diversity. There is no female. And we see along population that we have male gradients and therefore a genetic diversity gradient too. Here are, for example, the result of Valentine Chapelle, a former PhD in our lab, and she worked with 4 populations, 2 from Belize, 2 from Florida, and she worked notably with like the male ratio, the observed heterozygocy, and the selfing rates. And it turns out that here, Emerson Point preserved population in Florida have the lowest male ratio that is smaller than 1%, and therefore got the highest selfing rate of 97%. And that's the population I'm working with. So the materials method is actually quite classic. So I'm following Meaghan's article on optimal sample size to build an epigenetic clock, and I decided to rear 96 individual under standardized and controlled environments in the lab, and then I euthanize them at a specific time point between 60 and 1,300 days. Why these time points? I started at 60 because smaller than that, the individuals are too small and they have to pull the data of several individuals, and I wanted to have individual-specific data. And I went until 100, 300 days, which is a bit more than 3.5 years, basically because it's the mean lifespan of that species. So I have data that's covering the most lifespan of my species. Then we did DNA extraction from the brains using a nucleospin tissue exoskit. We prepared the library using the RRBS premium kit from Diagenode, and we sent the libraries for Illumina sequencing at the University of Liège. So RRBS stands for Reduced Representation Bisulfite Sequencing, and then it's a type of sequencing that's going to allow me to know the methylation data in my sequencing. So my treatment are going under a bisulfite conversion, where the unmethylated cytosines are going to be converted in uracils, and the methylated cytosines are going to stay as methylated cytosines. So when I have my sequencing data, I can simply map my reads to a reference genome and to a bioinformatically made reference genome that have a bisulfite conversion. And so with that comparison, I can have the methylation percentage of all my cytosines. So then it's a lot of bioinformatics to build the epigenetic clock. So first there is basically the preparation of the data. So I got a FASTA file from the sequencing platform, and I use notably TrimGalore to cut my adapters, FastQC to check the sequencing quality, and most importantly, Bismarck to, as I just explained to you, map my reads to my 2 reference genomes. Then I just explore a bit my data, mostly in RStudio using the package MetalKit, to have stats about the coverage and the methylation of all my samples. And finally, there is the model development. So here I decided to make an elastic net regression. Why? Usually when we would like to build a model that can predict things, so here predict the age of my individuals based on CPG, we either use lasso or ridge regression, but they both have the negative sides. And here the elastic net regression got a parameter that's called simply alpha, that got value between 0 and 1. When the alpha is equal to 0, we have a lasso regression, and when it's equal to 1, we have a ridge regression. And so here the elastic net is going to try 3 different values of alpha that are comprised between 0 and 1, and 3 different values of another parameter that's just the shrinkage parameters to see the importance of alpha. So basically we have 9 combinations of parameters, and the elastic net is going to choose the one that fits the most my data. To do so, I use the caret package in R, and I first split my data into training and testing data sets. That way, when I want to confirm my model, I don't have any bias here. And then I build the elastic net regression with first only my training data. first I did that with all the CPG that were sequenced and common to all my samples. And then I play a bit with the importance cutoff, that I will talk a bit more after, to only have a reduced number of CPG. And every time I was doing a new model with a reduced number of CPG, I always calibrate it and validate it on my testing data. So that's a bit what the informatics look like. So here, that's just to like split my data into training and testing. Here, that's how I'm actually making my model. So I'm using the train function, and I'm asking it to predict the age according to all the variables I'm giving it in my model. And I'm also doing some pre-processing of my data. So I'm like centering, scaling it. And NZV stands for near zero variance. So I'm removing the CPG that does not show any variance because they are obviously not going to be good predictor for age change. Sorry. And here, that's the type of result we have. So it's telling me the number of samples, the number of predictor. And you see the two first column, it's the alpha and lambda parameters that I was talking before. And then as a result, we have the root mean square error, the R square, and the mean absolute error. And we are going to choose the alpha and lambda parameters that got the lowest value for RMSE and NMAE, which is in this case the first one. And here, that's what I was talking about with the importance cutoff. So the importance of every CPG is between 0 and 100. So the first model, I took all my CPG, which is a bit less than 40,000. Then I simply put the importance cutoff at 1. So only 1 between 0 and 100, so it's really low, really to remove like all the nearly unnecessary CPGs that are mostly making noise in my model. And later on, I set up that importance cutoff at 35 in order to have really like the bunch of most correlated CPG to the age. So that's the type of result we're going to work with today. So we are plotting the predicted age against the actual age of my fish, and we have the training data in gray and the testing data in red. At the first glance like that, we can see that the dot fits actually quite well, so the prediction fits quite well the actual age of my fish. And that's actually also said by the R. So the R is the Pearson correlation parameters. And it's just a value that predicts, that's telling me if my prediction is correct according to the actual value. And so we have R that are really, really high, and we have p-value that are very highly significant. Also, we can see that the RMSE is quite low, and most importantly, that it's not significantly different between my training and my testing data set, meaning that we don't have any bias in the way I split the data. Here, it's when I put the importance cutoff at 1. So when I put it at 1, I'm losing actually most of my CPGs, or there are only 202 CPGs that like went up that importance cutoff of only 1. We can see, however, that the R and RMSE value are more or less the same or even better in that reduced data because we remove the noise of the CPG that were not really correlated to the age. And finally, we see here, when I put the importance cutoff at 35, I only have 23 CPG left in my model. We can see with the dot that it fits a bit less well than it was fitting with all my CPG or the 200 CPG. Still, the R value and the P value that are related to it are very highly significant, so that's very good result for only a group of 23 CPGs. There is also that PCA, so that's plotting all my individuals, and we can see on the first dimension of the graph, that's explaining 27.4% of the variation, that we have a clear separation of my individual by the age, with the very young on the left and the older on the right. No, it's the other way around. And we can also see on the second dimension that it's worth noting that the dots that are the most extreme in the second dimension actually fit to the individuals that are really young or really old. So, as a discussion, so we developed here the first epigenetic clock in a naturally isogenic vertebrate species. I would like to underline the fact that we really cover the main lifespan of the individuals, so from 2 months to 3.5 years, whereas in the literature, usually they focus on the really shortest period time, yeah. And as we said already, we have a very strong correlation between the prediction from the models and the actual age of the fish even when we only use a subset of 23 CPG sites. We have very good Pearson correlation and low RMSE value. And also another point is the mean absolute error. So that's parameters that's often given when we do epigenetic clock, and here it's of 100, 300 days. So it's a bit more than 4 months, which can actually seem a lot for age prediction. But as we have data from 2 months to 3.5 years, that's quite an acceptable value. And moreover, that value drops at only 102 days when we remove the oldest individual. We also have the median absolute error, that's only of 44 days. I also would like to underline the fact that I could not find any general hypo or hypermethylation of my CPG with the age. And so what's next? So as only a bunch of CPG are enough to actually predict in a good way the age of my individuals, we could develop a multiplex PCR. That multiplex PCR could be a tool, and we could see if we can apply it, for example, to other organs, notably the caudal fin, to develop a non-lethal method of predicting age that could actually be applied, for example, on the field, so to wild individual and wild population, because we don't have any information about the age of the wild population. After, we could also see if there is a conservation of that epigenetic clock through the different lineages of the mongrel revolus that have therefore different genetic diversity. And so we could study the independence of the epigenetic clock from the underlying genetics. Finally, we could see if there is a possibility of creating a general epigenetic clock for all the teleos, or there are more and more lab and articles published about epigenetic clock in teleos. Thank you very much for your attention, and I'm here for your question. So about the difference between sex, I'm only here using the hermaphrodite individuals. So I don't have sex difference because I only have like one sex in my individuals. Then I got lost in the second part of your question, actually.
Oh, so for the 23 sort of episnips that are variable over age, where are those sites? And are they?
I don't know yet. That's the next point I would like to work with, but I finished that like 3 weeks ago, so I did not have the opportunity yet. But I will look to that.
Excellent. Hi, everyone. Thanks for being here today. My name is Meaghan Clark. I'm a PhD candidate at Michigan State University. And today I'm really excited to share some updated results on a pretty big collaborative project I've been working on, looking at inbreeding in threatened rattlesnakes. So we know from a very large variety of systems that inbreeding can have some pretty dramatic fitness consequences. And we term this relationship where more inbred individuals have reduced fitness inbreeding depression. But inbreeding depression is really, really hard to detect in most wild populations. And this is typically done by using some sort of regression model between inbreeding and some measure of fitness. And while we can get pretty good estimates of inbreeding from genomic data, estimating fitness is still really difficult. And this is especially true in small populations of creatures with low encounter probabilities, which are often the populations that we care about from a conservation and management perspective. Additionally, a lot of proxies for fitness are only loosely correlated with true fitness, which makes detecting inbreeding depression even more difficult, even if it is actually present in your population. That being said, detecting inbreeding depression in wild populations is really important, not just for the conservation and management of those populations, but it also gives us really valuable information about how common inbreeding depression is across taxonomic groups and what aspects of fitness are most typically impacted by inbreeding depression. And so one way that I think is kind of exciting, an exciting way to kind of detect inbreeding depression by measuring fitness or inferring fitness is using long-term population monitoring data. And this has been done very successfully in a lot of really well-monitored populations like Florida scrub jays and Soay sheep, where these studies have shown that inbreeding really impacts a really wide array of fitness traits, from things like breeding success, juvenile survival, and annual survival. And so today I'm going to be telling you about how we're using long-term monitoring data to look at inbreeding and its effects in eastern Massasauga rattlesnakes. Eastern Massasaugas are a pretty secretive wetland specialist. They don't like to disperse through anthropogenic habitat types, so they're really impacted by habitat loss and fragmentation. And there's evidence that they've undergone recent bottlenecks. Eastern Massasaugas are currently listed as threatened or endangered everywhere they exist in the United States and Canada, and it's estimated that 63% of their historic populations are extirpated or are kind of on the way to being extirpated. So here we're using over 10 years of capture-recapture data to learn more about the extent of inbreeding in 2 different populations of Massasaugas, if there are fitness consequences, and also what factors might promote inbreeding within these populations. So these studies occur at 2 different Massasauga populations in southwestern Michigan, and these long-term monitoring projects are run by my collaborators, Jen Moore at Grand Valley State University, Eric Heilman at West Virginia University, and Lisa Faust at the Lincoln Park Zoo. Both these sites are fairly good habitat for Massasaugas, but they are surrounded by kind of a matrix of agriculture, so they're also fairly isolated. We have around 1,500 capture records of about 1,000 unique individuals from both sites combined. And from these samples, blood was taken from each snake, which was great for me. I extracted DNA and used a RAD capture method to get around 2,000 putatively neutral SNPs throughout the genome. We also have capture records and associated metadata with things like spatial locations of individuals and reproductive status. So from the genetic data, I estimated FGRM, which is a metric of inbreeding that's highly correlated with multilocus heterozygosity. 0 means no inbreeding, higher numbers mean more inbreeding. We also were able to use mark recapture models in the program MARC to calculate individual survival probabilities for each individual, and we were able to incorporate our inbreeding estimates directly into those models. We also reconstructed empirical pedigrees for each population using the SNP data with the R package Sequoia. And we used those to look at pedigree inbreeding as well as to measure reproductive output. So this here is an example, a highly simplified and truncated example of a pedigree from one of the sites. And these tiny little dots are individuals, and they're colored by their inbreeding value for that individual. And I know this is hard to parse, but the reason why I'm showing you this is because we can actually see evidence of close kin mating or inbreeding in the pedigree, and those instances correspond with our genomic estimates of inbreeding. So here we have 2 full siblings who were assigned to offspring in the pedigree, and that offspring was highly inbred. In general, we see that there are a fairly reasonable proportion of each population that show elevated inbreeding. Generally, anything above 0.05 to 0.1 is considered inbred. And so the big question here was, does this amount of inbreeding impact fitness? So first I'm going to tell you about our results for annual survival, and these analyses were actually done by my collaborator, Eric Heilman. So Eric developed a set of candidate mark recapture models, some of which included inbreeding as an effect to explain differences in annual survival, and some which did not. And we used AIC comparison to determine the most supported model. Our most supported model did include an effect of inbreeding on survival, and this effect was honestly shocking to us. So more inbred snakes have reduced apparent annual survival. So if you're a more inbred snake, you're more likely to not be in the population the following year. We were then curious if we would see similar effects in reproductive outputs. This was a little bit more complicated, in part because it's really hard to estimate age. So I really enjoyed the previous talk. If you're curious about how we incorporated that into the models, I'm happy to chat about that later, but just know that we are accounting for age to the best of our ability here. And we used zero-inflated negative binomial models. And our most supported model did include an effect of inbreeding on the number of pedigree offspring, but you can see this trend is fairly weak, and it's hard to know if this is really like biologically significant. But what is perhaps more significant is that we do see a pretty significant relationship between inbreeding and the probability of an individual having no offspring in the pedigree. So for each increase in FGRM of 0.05, that was associated with a 50% increase in the odds of having no offspring in the pedigree. Now, given the resolution of our data, we can't be sure if this is because, maybe inbred snakes don't have reproductive opportunities, they're not able to, get enough energy to reproduce. It also could be that these inbred snakes do have offspring, but those offspring, die before they are captured as part of this study. So we can't quite differentiate that here. But taken together, we consider this pretty strong evidence of inbreeding depression in these populations, and that inbreeding is impacting both survival and some aspects of reproduction. So that kind of led us to the question of why? Why is there even inbreeding in these populations? Both populations have census sizes of around 100 to 150, which is kind of huge for this species. So that this all was really surprising to us. And what we think is going on is that there aren't noticeable barriers within these sites. They're all pretty continuous, good habitat. But we do see this really fine-scale spatial structure. So these plots are the spatial locations of individuals based on capture records, and the individual colors are that individual's score on a genetic principal component analysis. I believe this is PC2. And so the different colors just to represent different genetic flavors. And you can see there's kind of some spatial clustering where a lot of the blue dots are close together and a lot of the green dots are close together. And we kind of see this fine-scale structure in the pedigree data as well, where close relatives like dam offspring and sire offspring, as well as parents who are assigned offspring in the pedigree, are significantly closer together than pairs of unrelated individuals. And what we think might explain this is actually the life history of Massasaugas. So Massasaugas have very, very small home ranges and limited dispersal. They also tend to return to kind of the same overwintering sites year after year. Males will often look for mates in the same locations, and neonates, young snakes, will often return to the area around where they were born to overwinter. And so it seems like there's kind of this spatial clustering and life history events in Massasaugas that might be generating this spatial structure, which is potentially also generating inbreeding. So to conclude, by using this really amazing long-term data set, we're able to not only quantify inbreeding in these two populations, but we're also able to determine that inbreeding is negatively impacting survival and reproductive success. And I think it's really exciting that we're able to find these results, even though we're not able to directly observe, birth and death events in these populations. We're still able to detect presence of inbreeding depression. And so I'd like to primarily acknowledge all the people who have been involved in collecting this data over the past 13 years. This is really hard field work. I'm very, very grateful to have had the opportunity to work with these people and these data. I'd also like to thank the Bradford and Fitzpatrick labs and lab affiliates, Lyle Gibbs and his lab for use of their reference genome and collaboration throughout the years, and funding from the Great Lakes Fisheries and Wildlife Restoration Act. And I think I have some time for questions. Thank you all. Yeah.
Have you looked into whether more inbred individuals tend to have higher natal site hatch rate and more outbred individuals?
That's a really great question. We unfortunately don't know where a lot of the overwintering sites are. We know where a few of them are, but these individuals aren't radio tracked or anything. So we don't have the resolution to answer that, but that would be really interesting. Yeah.
I was wondering if you can go back a couple slides to the spatial variation slide with lat long on the axis. Yeah. So it looks like there's a little bit of a different pattern of clustering with Berry and Cass counties, and like different amounts of that pacing to variation. Any thoughts as to why that is, or what it's referring to, and why there might be more inbreeding in one part or another?
We actually see like pretty similar levels of inbreeding at each site. I think this is like a little anecdotal. I haven't like done stats on this. I think in general the individuals in Cass County move around a little bit more. Kind of like the maximum dispersal distance between capture events is a little bit longer. But that also might be due to differences in sampling efforts between sites as well. Good question.
I'm not that familiar with the FGRM metric, and I was wondering if you could just explain it a little bit. Like, is it relative to the genetic background of these two populations?
Yes, so it is relative to the allele frequencies within each population. So it's mostly capturing the effects of, or it's mostly measuring kind of the amount of what I consider like true inbreeding, so non-random mating within a population, rather than the effects of drift. So we have not yet quantified like, you know, how much drift has impacted either of these populations and if that might have fitness effects. All right, thank you.